Okie dokie. Hello, everyone, and welcome to another GlycanAge webinar. Um, Thank you, everyone, for turning up. I'm sure we're going to have some people joining the call. For those of you who do not know me, my name is Dr. Simi. I work with GlycanAge in the business development side, but also on the educational side, hence why we're doing this webinar today. And the reason for the webinar, of course, is because I think this topic is one of interest in particular. I've had quite a few partners that we work with having lots of really interesting discussions about the different biological age clocks, how they are different, and actually how do you determine which clock is the right clock. And so in today's webinar, I'm going to be speaking about a few of the biological age clocks. There are so many, and so I'm going to be speaking about three in particular. And so one thing I would ask is, as always, please do ask questions. You can put questions in the chat box and we will answer them as we go through. And feel free to, if at the end, I'm probably going to ask if there are any other topics. If there are any clocks I don't speak about that you're interested in knowing about, we have a whole PhD team behind us who knows a lot about this stuff and does a lot of research. So definitely share that with us as well. But happy to just jump in. Hopefully you can all hear me. I hope. I'm going to take that silence as a yes. All right, awesome. So biological age clocks, let's talk about what they are and why they're so important. And all of you can hear me, which is great. Thank you, Adrian. Okay, so I think the first thing we need to discuss is what is aging. And I think this is a really it seems like a very simple question, but it's actually a very difficult question to answer. And the reason for this is because aging is multifaceted. There are lots of different ways that one can age. But one thing is very, very clear is that aging is the biggest risk factor of chronic disease. And that is one of the reasons why it is such an area of interest. But when we think about aging, I think we can categorize it into two very distinct types. So you have your chronological age. This is what I like to call your passport age or your driving license age. And that is simply the amount of time that has passed from birth to whatever given date. Most of us age chronologically the same. Whether you want to lie about your age or not, the fact is we're all aging the same when it comes to chronological age, and this is non-modifiable. But when we speak about the other type of aging, which is biological age, we know that this is actually different. And this is more about your physiological age. It's a measure of your physiological health, your functional capacity. And actually, we do not all age biologically the same. And some people age faster than others. And this can be affected by so many different things, genetics, by your lifestyle and various other things. But ultimately, when we think about biological age, what we know is that it is modifiable. And that is why it's a particular area of interest, especially in this longevity space that we're all part of. So when we think about biomarkers and we think about biological age is typically calculated or a biomarker is used to determine what a biological age is. Now, if we think about what does a biological age what does a biomarker need to be in order to be successful? There are lots of different categories and criteria for this, but the one that's probably the most popular is the one that was put together by the American Federation of Aging Research. And what they ultimately said was that in order for a biomarker to be ideal, it should predict future health and survival better than chronological age. So you're looking for something that is a better prediction of what your future health and survival is. It should also monitor a basic process that underlies the aging process. Now, as we've mentioned, aging is very multifaceted and has lots of different elements to it. And so actually there are lots of different processes that underlie the aging process, which is why you have so many different biomarkers, which is hugely interesting, but can be really, really confusing. The other thing is that you want it to be repeatable without harm. So if I have to do a liver biopsy every time to tell someone what their biological age is, that is not a good thing, considering that, liver biopsies are actually high risk procedures. And so you want something that is easy to do, doesn't cause too much harm. I think a finger prick is enough, but we don't want to cause any more harm than that. And then this is one that kind of is mentioned by the American Federation of Aging, but also in other texts as well. But this concept of being both reproducible and responsive. And what we mean by this is that if I do a biological age test for someone today, I want to be able to do the same test tomorrow and get the same result. But also, if I implement a number of different interventions, I want to see that that biological age will respond to those interventions as well. And I think that's incredibly important when we think about why biological age is necessary, is because we actually want to see what interventions work for individuals to reduce their biological age. And then finally, we want it to work in both humans and laboratory animals as well. And the reason for this is because, of course, with biological aging, you would want to do tests to understand actually how do interventions affect that. And actually, if it works in both humans and laboratory animals as well, you can do that testing as well. And so these are the 5 things that much of the research and the papers talk about when they're looking for an ideal biomarker. So I did a lot of sort of reading and digging and looking at the research in the space, and there is a lot of information out there, and it can be a little bit confusing, especially when it comes to biomarker categories. There's lots of different ways that you can categorize biomarkers, but here I've just chosen what I would consider to be the main categories. And so first of all, you have these histology-based markers. These are these markers that tend to be focused on your cellular, molecular, genetic data. So in that category, you have your epigenetics, so your DNA methylation, you have your metabolomics, your proteomics, and your glycomics, all these different areas of study. And then you have the clinical biomarkers. If you're a clinician or a practicing doctor or someone who works in the space of medicine and health, then this would be your typical blood chemistry, so your hematology, your liver function tests, those kind of things. But also in this space, you also have anthropometry, which is a really difficult word to say. But ultimately, this looks at things like your BMI, your hip waist ratio. All of these things can actually be used to create a biological age. And then finally, you have what I'm just calling the others, kind of a mixed match of very interesting novel research, which is looking at things that are like image-based. So they're now tools that you can use to measure face aging, even ECG aging, and also MRI scans, which can actually give a brain age as well. And so these are more novel, but there's a lot of research in this space. And then finally, you have what I'm calling composite aging clocks. And I would say these are sort of the next generation clocks. They're the ones that are based on AI. They bring together, all of your big data, all of your wearable devices, to create kind of like an age that's based on that person's information, as well as some of the histology based markers that we're speaking about, and also the clinical biomarkers as well. So, as I mentioned, there are a lot of clocks that exist. And what I've done is I've looked at the ones that I think are the most popular, the ones that have the most information on them. And I'm going to speak about 3 in particular. So there's going to be epigenetic clocks, which I think is the one where there is the most research and people know the most about. And what I will do is I'll break these down, not just into types of clocks, but actually different organizations that do provide these clocks. So for example, True Diagnostic probably being a very big and well-known provider of epigenetic clocks. And then we'll speak about glycomic clocks as well. Are we a bit biased? Maybe, but I'm going to try and give you as much of a neutral opinion on all the different clocks. And then finally, we'll talk a bit about proteomic clocks as well, which are becoming more popular, especially in the research space. Do we have any questions so far? It doesn't look like it, but if anyone has any questions, feel free to just drop them in the chat. Sorry, my headphones are being a bit funny, so if anyone has any issues with sound, please do let me know. Okay, so first and foremost, the epigenetic clocks. Now, these are, of course, I think, the ones that people know about the most, and they're based on these chemical changes that affect gene expression, which occur predictably over time. Typically, it's based on DNA methylation, but it can also be based on other things like genetic change as well. And there are many commercially available epigenetic tests, but the ones I'm going to focus on are probably the most popular ones, which are your true age clocks. Those ones that, your true age clock, the ones that are based on the Steve Horvath based clocks. So that's your DNA age and Grim age. And then also I'm going to speak briefly about the Levine based clocks, which is your pheno age as well. I think we have this question. So someone's just asking if there'll be a recording. Yes, if you signed up to this webinar, please note that you will get a recording of the webinar, and it will also be available on your dashboard if you're signed up as a partner as well. So don't worry if you've missed the beginning, not to worry at all. Okay, cool. So let's get into it with epigenetic clocks. So just wanted to give a brief on the science. I think we have quite a different a broad group of people on here. Some of you may be scientists and know far more than I do about epigenetics, but I'm going to give sort of a very brief overview and then speak about a bit more deeply about the pros and cons of the different clocks that exist. So when we're speaking about epigenetic clocks, I think there's three very important things that you need to understand and you need to know about. The first is CPG islands. So these are basically regions within your very complex DNA where you see a cytosine and a guanine nucleotide, and they kind of repeat. And these are a sort of repeat in a linear sequence. So these are what you call these CPG islands. And then you have methylation. So methylation is the process of adding a methylated group onto that area of DNA. And typically that changes the way the gene or that genetic information is expressed. And then finally, you have DNMT, which is ultimately these enzymes which facilitate that methylation process. And so these are the three important features when it comes to thinking about epigenetics and what's happening in the body. Now, as I mentioned, I'm not going to go into great detail about, we're not doing a class on epigenetics here. I'm sure many of you would love that, but I'm not going to be doing that today. But hopefully that gives you kind of some brief understanding of what epigenetics is and the three very important elements of epigenetics. So then when we think about the sort of more science behind epigenetics, what we know is that you have these CPG islands, and when these islands get methylated, so when you add this methyl group onto these areas of DNA, it promotes gene silencing. So ultimately that gene kind of gets turned off and it stops being expressed. It's a dynamic process, so it is not just like the gene turns off and it's always switched off. It can be switched on and off, and all of that is controlled by these enzymes, which are called DNMT. And then that DNA methylation, when it happens in certain CPG islands, tends to increase in certain areas with age. And actually, when we think about the first generation of epigenetic clocks, a lot of them were actually based on these age-related changes that happened kind of chronologically with age. And so a lot of the initial or first generation of epigenetic clocks were very, very good at estimating chronological age. But if you remember what I said about the criteria of what an ideal biomarker is we don't want it to estimate chronological age. We want it to estimate biological age. And actually, if you have gene areas that are specific to chronological age, that actually is counterintuitive because it's not really giving you biological age information. It's giving you chronological age. And so that's a really important thing to remember, because actually one of the challenges with epigenetic clocks is understanding how has this clock been created? What CPG islands is it actually looking at? And this information can be quite hard to elicit from different companies. And I'll speak a bit more about that when I talk about the specific companies that provide epigenetic clocks. But typically with these companies that provide these clocks, they will use regression modeling, statistical models, mathematical modeling to identify CPG islands, so these areas that are associated with age or morbidity or mortality, so death or sickness. And then they'll weigh them based on their importance with aging and use the algorithm to come up with a biological age. And so there's lots of different elements when it comes to epigenetic clocks that are combined together to sort of create or to calculate a biological age. I'm just checking, I think we have something in the chat. Okay, cool. So just in terms of, to conclude on sort of the basic science of epigenetic clocks, what we know is epigenetic clocks are powerful tools. I don't think that can be denied, and they can be used to measure biological age. There is a huge amount of variation in how genetic clocks can be modeled. And the reason for this huge variation is because of all these various CPG sites that can be used. And one of the biggest challenges in epigenetics is identifying which of these CPG sites is the best to measure biological aging and not just chronological aging. And so as a tip for any of the clinicians or scientists out there who are working with patients or doing research, I think it's really important that when you're using an epigenetic clock, you're doing your due diligence. You're understanding what the data set was used, what data set was actually used to create that clock, and how the CPG sites were chosen. Were they chosen based on areas of genetic information that are affected by inflammation? Was it based on liver health? And these are various different sites that you can use to identify different areas of mortality or morbidity or of chronological age. The other important thing to remember with epigenetic clocks is because they are typically modeled, they are typically based on some assumptions as well. And so as epigenetic clocks and companies use these algorithms, actually, as more information becomes available, those algorithms can change. And what this means is that if you're using an epigenetic clock and they change their algorithm, and you have a patient and you do their test 5 years ago and then you do it today, if they've changed the algorithm, actually the calculation is slightly different. And so it can be hard to compare over time how someone's epigenetic age or biological age calculated from the epigenetic clock, how it's actually changed because of those changes in algorithms. And I think this is something that is missed quite a bit, because I have spoken to partners who have been using epigenetic clocks and they notice that the algorithm changes. There's an email that says the algorithm has been updated, but if you're not aware of that, then actually you might get slightly skewed results that don't necessarily make sense. So definitely something to be aware of when it comes to using epigenetic clocks in practice. I've listed the sort of pros and cons there, which I'll go through very quickly, but I think we've spoken really about those. But largely speaking, some of the pros is that because there are a large number of CPG sites, you can profile, you can improve accuracy over time. And as more research is done into epigenetics, I think we will see an improvement in the accuracy of these clocks. But also the clocks can be trained against so many different things. And so they can be trained against clinical data, they can be trained against different CPG sites, and so on and so forth. And so you can really get a lot of information out of epigenetic clocks. However, there of course are cons. I think accuracy is a big one. So the accuracy of an epigenetic clock is very highly dependent on the company that is doing it and the algorithms that they're using. And so actually, not all epigenetic clocks are created equal, and actually you can get variation across accuracy with them. But also, as mentioned, the different clocks can use different CPG sites. So if you do an epigenetic test with one company and you do an epigenetic test with another company, actually they could be using completely different elements of epigenetics. And so the results can be very, very different. And then, as I mentioned, because they're based on this modeling, there are assumptions that are being made. And typically one of the key assumptions that we typically see in research is that actually the assumption is the rate of methylation or the change in methylation is constant over an entire lifespan, which of course is not true. The way that you age between the ages of, 1 year and 20 is very different from how you age between the ages of 60 and 95. And so again, there are these assumptions that are made that you have to consider when you're using these clocks. Great. So one thing I did want to do, as I mentioned, was to be focused on both the science, but also the commercial aspect as well. If anyone here is a researcher, obviously the number of clocks that are available in the research world are very vast. But if you're working in clinic or you're a practitioner or you're an individual who is using clocks, then actually the commercially available ones are the ones that are probably the most important to know about. And so what I'm going to do now is I'm going to talk through some of those commercially available clocks and go through some of the pros and cons of them as well. And as I mentioned, if anyone has any questions, please do feel free to just drop them in the chat. I hope I'm not talking too fast. Okay, so I think the first one we have to talk about is True Diagnostic. I'm sure many of you are aware. Sorry, someone's just asking in the chat, what is a CPG again? So I did discuss this slightly at the beginning, but ultimately a CPG island is a space within your DNA structure where you have a lot of cytosine and guanine. So these are repeated sequences, and these are islands that exist in the DNA structure that change quite predictably as you age and as you are exposed to disease. I hope that answers that question. So when we think about True Diagnostic, so True Diagnostic is a very large and reputable company. They are focused on epigenetics, and they have two main products, TrueAge Complete and TrueAge Pace. So the test retails for about $499 per test, and it is based on a whole blood sample, which uses a sort of blood collection sample. So you do a finger prick and then you would do a few drops of blood into the collection bottle. But they also use the Tasso device, which is a device that goes on the arm and extracts blood from you as well. And so they have those two methods of collection. Now, the TrueAge Complete product provides information on 3 different areas. They provide information on DNA methylation, they provide information on the telomere shortening, and also on the pace of aging. I'm going to speak specifically about the pace of aging in the next slide, but in this section, I'm just going to focus mainly on the DNA methylation. So when you get a TrueAge diagnostic report that is part of TrueAge Complete, they will give you data on how alcohol intake might affect your gene expression, how the body will respond to caloric restriction. So for anyone who is focused on that area or reducing their caloric restriction, how their body might respond, but also how smoking has affected gene expression as well. And I think we can all agree that that's, quite useful information that one might want to be aware of. In terms of how the test is done, so this information was taken from their website and also from some of the research papers that they have on their website as well. So it's based on 850,000 locations on the DNA. Now, I looked and I searched and I couldn't understand exactly what that means. So we spoke about these CPG islands that we know epigenetics is focused on, but actually, when you say 850,000 locations on the DNA, I'm not sure exactly what that means in terms of the DNA. Is it 850,000 CPG islands? I don't think so, but maybe it's just the specific gene areas or specific bases. And so this just kind of, I guess, gives that indication that sometimes finding information when you're looking at specific companies that provide epigenetic clocks can be quite difficult to understand what the clocks are based on and exactly what the clock is looking at. But when we look at the research, they do have a research base, and the database is of about 5,000 people that they did testing on and validated these clocks on. And in terms of the benefits that you get with True Diagnostics, so having looked at one of the True Diagnostic reports myself, I would say it is a very comprehensive report. There is a lot of information that is included within it. The information provided can be really useful in supporting clinical decision-making for patient health optimization, but also, True Diagnostic is a really well-known brand and it's also widely available. Sorry, someone's just asking. So yes, everyone, just a reminder, the recordings will be sent to everyone after the webinar. So don't worry if you missed the first half, no worries. And then in terms of limitations, so we've spoken a bit about some of the limitations, but I think when it comes to commercially available clocks, it really, for me anyway, when I was doing the research for this, it is really hard to find the scientific backing of the clock. So we know epigenetics. Epigenetics is a very well-known field. There is a lot of information about epigenetics, but looking specifically at company clocks, it can be difficult to understand, what CPG sites or what what parts of genetics is this clock based on? And I think that can be quite challenging. And as I mentioned before, one of my key tips if you're using an epigenetic clock is you need to understand the data set that it's based on, but also the CPG sites they're looking at. Because what you don't want to do is pick a clock that is measuring chronological age as opposed to biological age. Also, models used to calculate the age are subject to change. And I think we've spoken about this already, but that makes continuity quite difficult. So if you're working with people and you're testing them over various time points over years and they change the algorithm, again, it's going to change the output. And so that's something to consider as well. And then with telomere shortening, so the TrueAge Complete does provide information on telomere shortening, but it is quite well known in the scientific space anyway, that actually telomere shortening, while useful, is a really challenging measure of biological age. And the reason for this is because the value of the information provided from telomere shortening is really, really heavily dependent on the sample. So just to give you guys an example, if I was to take a blood sample today and I got a bunch of cells, and I look at the telomeres. So just for anyone who doesn't know what telomere is, that is the sort of, I guess, the cap or the end of DNA that shortens over time. If I take a sample and I measure the telomeres of all the cells in that sample or the genetic information in that sample, and then I take another sample from a different area of the body or from a different tissue, actually the ages that I get back could be completely different. And typically they will use models and use algorithms to kind of estimate the whole thing, but it is quite well known in the scientific community that telomere shortening can be really hard to actually draw information from because of that variation that you can get within tissues. Great, I'm just seeing a question here, so I'm just gonna get that. Yeah, so someone has quite helpfully said, in the chat that actually when companies do revise models, this person lost about 5 years. And so, it really is challenging when you have companies that are constantly updating these algorithms. And this is one of the big issues when you have a mathematical model-based biological age, because it's not just the it's not just looking at the sample you give, it's also being applied to a model. And if that model is changing, then yes, you might add or lose years. And actually knowing what's happened can be difficult, especially if you don't actually know that they've updated the algorithm. Thanks for that, Bruce. Okay. So now I'm just going to talk a bit about TruePace. So TruePace is actually, to me, is actually really, really interesting. And it's interesting not because of TruePace in particular, but because of the algorithm that it's based on, and the research that they did, which I think is really, really fascinating. And if anyone is, a really budding researcher and loves to read papers, I would really recommend having a look at the paper. It's a really quite a unique study that they've done, and I will sort of give the sort of bullet points on it, but it's quite a fascinating study in that they've used a cohort study. So they followed people over time and they've looked at how their organs have aged. So TruePace is another test that's offered by True Diagnostic. It costs about $229 for the test. And in this test, you typically get your telomere length, and also this pace of aging, which is again calculated based on the model. Now, the pace of aging data is based on the Dunedin pace study, which then created an algorithm. And this algorithm was based on this population of 1,037 babies that were born between the years of 1972 and 1973. All of them were born in Dunedin, which is a place actually in New Zealand, hence the Dunedin pace. And what they did is they looked at 19 indicators of organ system health, and they did this over 4 different time points over 20 years. So they looked at their values at age 26, 32, 38, and 45. Now, the 19 indicators, I haven't listed them here, but they did look at 7 different organ systems. I can't remember exactly all of them off the top of my head, but it was cardiovascular health, metabolic health, immune health. They also looked at lung health, and there were 2 more in there, which I will surely remember shortly. But they basically looked at all these different organ systems and then used the different values in the organ systems to create an algorithm of how the different organ systems age, and then put this in a mathematical model to understand actually how someone was aging over time over this period. So it really is, I think, a really interesting study. But the real question is, how does this match up with actual biological age, right? Because that's what we're all interested in. We want to know, actually, if I'm looking at different organ systems, how do they age? So I think from my perspective, the Dunedin study in and of itself is very good, apart from the fact that the sample size is quite small. So yes, they looked over a long period of time, but the population size is actually only 1,000, which is actually a relatively small sample size. And so it does potentially call into question the validity of the results, in particular because the population they used was not very diverse. So I think it was 94% white. And they actually only looked at changes up until about the age of 45 and then used the mathematical model to extrapolate the rest. And I think, as we've discussed, aging is not linear. It doesn't happen in a straight line. It changes as you age. And so as you age, typically what you find is your pace of aging or the way that you're aging does change. And so one question I definitely had was actually how have they predicted or modeled for that in the algorithm that they built? So it's definitely an interesting study. It's definitely an interesting way to look at aging. And actually, we were at a conference not too long ago in Israel, where we had Professor Andrea Meyer, who is someone who is a medical director of quite a big longevity center in Singapore. And she did speak quite a bit about how aging can affect various organs and actually how they relate to one another, and that you see that there is relation between all the different organ systems. But again, one of the questions I have is actually, if you're looking at 7 organ systems, how do you determine what proportion each of those systems contributes to aging? And these are all the questions that I think it's really important to ask when we are choosing which clock we're going to be using. If you're using a Dunedin pace clock, actually, how do you know what part of an organ system or how much of an organ system affects the way someone ages? Because actually, what we do know is that some people have a more resilient liver than others, for example, and some people are more susceptible to forms of liver cancer. And so actually, how does that translate into the algorithm? And for me, this is one of the biggest issues that I find when it comes to epigenetic clocks and understanding how the algorithms work. And I don't think it's necessarily an expectation that as a clinician or individual that you have a full understanding that effect, but I think it's actually on the company to give that information and to share that. And one of the things that I did struggle was to actually extract some of that information from the TruePace website in terms of actually what does this mean in terms of if my liver is aging more, how is this going to affect my biological age, and what percentage of an effect is it going to have in the age that they give me? So these are definitely things that I think it's important to consider. But overall, I think that the Dunedin pace is incredibly interesting. The key benefit is that they used a cohort study, which is very unique, especially in the longevity space, which is still relatively new in some ways, and actually were able to look at people over time. They used really valuable endpoints. So they looked at things like cognition, physical function, facial aging, and subjective signs of aging. So ultimately, how did those people individually feel that they had aged as well? And also they were able to show sort of high test, retest reliability as well. And then also there was good correlation with other biological age markers as well. So I think these are all very positive. In terms of limitations, I think one of the key things is that it assumes that aging represents progressive decline across organ systems. Now, actually, there is a lot of research to show that this is the case. And so I don't think that necessarily is a limitation. But what I would say is that it is difficult to understand how much an organ system contributes to aging and how they've actually worked out within the algorithm that they built. Also, again, as I mentioned, the population diversity was quite limited because it was a study of only 1,000 people. Also, one of the limitations that they do actually mention in their study as well is that it's not yet known if the Dunedin pace is modifiable by interventions. And so I think one of the things we discussed at the beginning was that biological age is useful because we want to understand what interventions can I put in place for my patient or for myself that will help me to reverse or to slow down my aging. But actually, if you have or you use a biological age clock that isn't modifiable with an intervention, what you ultimately are saying is that the biological age is fixed, which we know is not the case. And so as the group that did the Dunedin pace research continues to do research and publishes work on this, I think it will be really interesting to see if they see that the age is modifiable with interventions as well. But at the moment, that is not yet known. And so it kind of creates a question of how useful is this if I don't know if it's going to change when I make interventional changes. And then finally, as I mentioned, they actually only looked at the biomarkers up to the age of 45, but we actually know that age does change as you age, your pace of aging and the way you age changes as well. And so there's even more scope to do further research to understand how people age beyond that. And yes, they use the mathematical model, but we've already discussed some of the limitations of doing that kind of research. Does anyone have any questions on that? Because I feel like the Dunedin pace one is one that people have asked me a lot about on calls. And so if people do have questions on it, please do feel free to drop them in the chat rather than waiting until the end. Not yet. So either I've confused people so much that they don't understand what questions to ask, or all of you just know so much about it already that it doesn't matter. Okay, that's fine. Let's move on. Okay, so yet another epigenetic clock. As I mentioned, there are quite a few of them. So these are Horvath-based clocks. And when I say Horvath-based, I'm sure many of you know the name Horvath. Stephen Horvath is considered to be almost like the father of epigenetics, did a huge amount of research initially and was one of the people that created one of those initial first generation clocks, which were very, very good at measuring chronological age. But then there were questions about whether it was good at measuring biological age, which is of course different. So there are many companies that provide Horvath-based clocks. MyDNAge is one of them, and their test costs about $299. Similar to TrueAge, it is an at-home whole blood sample using a collection tube, provides information on DNA methylation, and it's based on Stephen Horvath's original clock, which looks at 353 age-dependent CPG sites. So, you know, it is looking at a huge breadth of information. But again, as I mentioned, what's really important about epigenetic clocks is not that you look at CPG sites that are just age dependent, but actually ones that are dependent on biological aging. And so actually, Stephen Horvath has continued to do a lot of work in this space and continues to do a huge amount of research in biological aging. And one of the newer clocks that they have created in their scientific research team is GlycanAge 2. And so this is an upgrade of the clocks, GlycanAge 1 that already exist. And ultimately what they did with GlycanAge 2, and again, sorry if I'm getting too much into the science, but what they did with GlycanAge 2 is they ultimately looked at 7 DNA methylation based estimators of plasma proteins. So just to explain that a little bit, because it is a bit hard to get your head around, but they identified plasma proteins that were associated with disease, illness, death, and sickness. And then what they did is they looked for CPG sites, so areas of methylation that were associated with those plasma proteins. And then what they did is they used those sites to calculate what the biological age would be based on the level of those plasma proteins in the body. So again, with epigenetic clocks, there's quite a lot of steps, and they're quite calculated. But ultimately, what they did find is that significantly improved the way that the biological age clock was able to predict various different time points. So in the case of GlycanAge 2, they used the time point of time to cancer and time to heart disease, which of course are very interesting and useful endpoints to look at to understand how effective or how useful a clock is. Because as I keep mentioning, the clock is only useful if it's going to give you more information about your health level than your chronological age. And so not only do they look at these plasma proteins, but they also look at your smoking pack years. So it's, I think it's very well known that smoking has a huge impact on your longevity. And they also looked at chronological age, so at the time of the sample, and the sex. They combined all of this information, stick it into a machine learning algorithm, and they spit out a biological age based on the algorithm. And so this algorithm is validated. So they validated it using a number of different samples on individuals, about 7,000 individuals from the ages of 40 to 92. So, and looked at various different ethnic groups, and that's how they actually validated that the algorithm they're using is working. And I think some of the key benefits, as I mentioned, is that they use really valuable endpoints. So time to cancer and time to heart disease, which I think are things that are very useful to understand. But actually, when we think about these kind of clocks and the GlycanAge clock, really useful for research. But actually, Stephen Horvath himself has said that from a clinical point of view, or from using this within clinic for patients, it's not quite there yet. It's not clinically useful. And the reason for this is because of the large error margins, potentially because of the calculations that are needed, but also because the clock ultimately assumes that these 7 plasma proteins that it's focused on are the best predictors of mortality and morbidity, which is, of course, not necessarily the case. And it comes back to this point of research that actually there is more research needed to understand which which CPG sites are the most useful in estimating biological age. Finally, I would say that when it comes to Horvath-based clocks, there are quite a few commercial companies that claim to be based on the Horvath clock, but there's this lack of transparency on what elements of the Horvath clock are these companies based on. It's not always very easy to see what part of the Horvath clock have they taken, what part of the algorithm have they actually used. And again, you have this question about how do you know what algorithms and what sites and what information they're building this biological age on. And so again, it's about doing that due diligence and understanding actually, is this clock really based on science, or how much of the Horvath research base are they using, and how useful is that for my patients and in practice? Any questions? No, I've shocked everyone into silence. Good, okay, great, I'll carry on then. Finally, this is the last epigenetic clock I'm going to speak about, so we will move on. We have the Levine clock. So Levine, again, is another really big name in this space. And the clock associated with Levine is the PhenoAge clock. So this is probably the cheapest clock that I'm aware of, especially in the epigenetic space. Costs about $75. Then lots of different people who provide this test. Similar to all the other epigenetic tests, it requires a whole blood sample, so a little test tube again. And then it provides information on DNA methylation. Unlike the first generation clocks, which I spoke about, that looked at the CPG sites associated with chronological age, this actually tried to look more at biological age. And the way in which they did that is they used both the chronological age, but also 9 clinical biomarkers, and they used this to create this kind of phenotypic age. And they also looked at 513 CPG sites. So again, looking across the DNA, which areas are associated with changes in age. And that is how they created this PhenoAge. And it looks at 9 clinical biomarkers. I'm just going to put them on the screen for people who are really interested. So for many of you who work in sort of as physicians or in the space of medicine, these will be things that you recognize. So albumin, creatinine, glucose, your CRP, your lymphocytes. So this is your white blood cell level, and your red blood cells and things like that. And so what they did is they looked at these 9 different clinical markers, combined this with the epigenetic CPG sites, and then use that to create a model that then spits out an age. Now, again, I think this is incredibly useful for research. It correlates very well with existing clinical parameters that many physicians or clinicians would be aware of. But again, potentially higher margins because it's a derived value. So rather than looking directly at the level of those markers, it uses the CPG sites to estimate what those biomarkers would be, and then create a biological age from that information. And so again, you have this questionability on how clinically useful is this, when actually, why would you not just measure the clinical parameter itself? And I guess that this might be why this is a slightly cheaper test than anything else. But it is just interesting that they use the CPG sites to estimate the clinical markers rather than just measuring the clinical markers directly, which is a question that I had when I was doing the research for this. And then I think one thing also is, again, aging is really complex. There is no single biomarker that can tell you very clearly what someone's biological age is. This test assumes that there are these clinical parameters that can be used to calculate an age, and that these parameters are the best predictors of morbidity and mortality. But again, this is not something that we can say for sure. And so again, it's all about adding to the research and using that research to identify which areas or which parameters would be best. Awesome. Okay, so that's epigenetic clocks done. This is why I could only pick 3 clocks, because obviously there's so much information in them. But now I'm going to talk a bit more about glycomic clocks. As I said, obviously we are GlycanAge, and so I'm going to try and be as neutral as possible, but certainly this is an area that we probably know the most about. So when we think about glycomic clocks, the first question is, what is glycomics and what is it based on? Glycomics is, even though it's been studied for a very long time, I think many people don't know very much about this area. And so we're trying to do a lot in terms of education and to help people understand this area, because I really do believe that glycomics is like this, hidden treasure that many people don't know about. But ultimately, glycomic clocks focus mainly on these complex molecules called glycans. Glycans they exist on every cell surface in the body, and they enable cell-to-cell communication. So on the right here, you can see a picture of a cell, and all these orange things on the outside are the glycans. They develop together with proteins and lipids, and the glycans are what make proteins functional. And also when we think about the human glycome, it's actually adjusted and altered by your genetics, your epigenetics, and your physiological health. And so actually all of these things can have an effect on the glycans that you have on your various cells. When we think about the commercially available examples, GlycanAge actually, as far as we're aware, is the only commercially available glycomic clock. And so that's obviously very useful, because it means that we have a huge amount of research and information on glycomic clocks. When we think about the science behind IgG glycomics in particular, so I've spoken about glycans and the human glycome. The human glycome is a collection of every glycan you have in your body, of which there are billions, because they are literally on every cell in your body. Typically, when you're looking at research, they're focusing on IgG. So IgG is a functional protein. It's part of your adaptive immune system. And your adaptive immune system is the part of your immune system that literally learns. So as you're exposed to infection and disease, your adaptive immune system learns and kind of stores these memory proteins. And IgG is an example of one of those memory proteins that then would fight the disease if you were to get it again. When these IgG proteins fight infection, they actually learn as they do it, and they kind of create a memory. And this memory exists on the surface of the IgG in the form of glycans. And when we think about the IgG glycomics, that is what we're looking at. So we're looking at the glycans on the surface of these proteins that are sort of your immune system fighters. And these glycans change both in response to disease exposure. So as you're exposed to these diseases and they're fighting these diseases, these glycans will change. They change in response to inflammation. They also change in response to your physiological health, genetic risk, and also epigenetic changes in your gene expression. And so within that glycan, that language that is on this protein, there is a huge amount of information, and it can really help you indicate the inflammatory state of the immune system. And that is what glycomics is looking at in order to determine your biological age. Okay. So when we think about glycomics, the reason why it's useful is, as I mentioned, you have these glycans on the surface of these really important proteins within the body that help fight infection. But actually what we know is that as your body is exposed to more inflammation, more disease, these glycans change shape, and they actually become pro-inflammatory, or they become anti-inflammatory, depending on where you are on that spectrum of disease. And so depending on your physiological health status, your genetic profile, these glycans can become more or less pro-inflammatory. And this diagram quite nicely shows just how these glycans can change as you're exposed to more inflammation or less inflammation. One thing I did want to mention very, very briefly was glycocalyx. Some of you may have heard this term before. I had a partner who we were on a call and they mentioned glycocalyx, so I thought it'd be useful to just mention it here. Glycocalyx is sort of under the umbrella of glycomics. Your glycocalyx is ultimately all the glycoproteins and glycolipids, so all these structures that surround your cell membrane and bacteria and all the various cells in your body. In particular, glycocalyx of recent has had a bit more buzz around it because it's being used specifically in cardiovascular medicine. So people who look at your vasculature, so your blood vessels. And what we know now is that actually in your blood vessels, if you imagine your blood vessel as a tube, the inner surface of your blood vessel has this kind of glycocalyx layer, which is this layer of these complex molecules. And actually what they're doing now is they're analyzing what that layer looks like to give information about someone's cardiovascular health. And so glycocalyx is under the umbrella of glycomics. So for anyone who's heard that term, it is under the same umbrella, not exactly the same, but it is a study of those very important molecules, which are called glycans. And actually we are seeing a lot of clinicians, especially those kind of cardiologists and people who are in that space, using and learning more about glycocalyx now. And this is what I mean when I say glycomics is like this untouched treasure. I think there's so much information within the glycocalyx and within glycomics that is still to be discovered. And so there's a huge amount of potential in this research area. And actually one of the things we know now is that as you age and as you have an increased risk factor of different diseases, your glycan pattern, so the types of glycans you have on your cells, on your IgG, can change and can give you an indication of what diseases you might be at high risk for. So I think just to kind of summarize everything I've said, the glycans that we have in our body provide a wealth of information. The challenge is organizing that information into a way that is easily understandable and that can help people understand what their biological age is. And that's ultimately what we do at GlycanAge. What we know is that when cells become senescent, so senescent is what I call zombie cells. So they're cells that no longer function, but they don't die. They stay present. And what happens is these senescent zombie cells release these inflammatory cytokines. And sometimes that can be a good thing, but sometimes it can be a very negative thing. And it causes a bunch of responses that can be quite bad for the body and increase chronic inflammation. But also as you get these necrotic or damaged cells, you get release of various other indicators, which all contribute to immune dysregulation. Immune dysregulation leads to inflammaging. Inflammaging changes your glycans, and therefore by analyzing your glycans, you can understand all of this process. And so you can really understand how much information is packed into your glycans. And this is why, for me, I think it's a really fascinating area of study. And we at GlycanAge are trying to leverage this to give people the information they need about their disease risk, but also about their biological aging as well. I see we have a question. How can antioxidants such as various vitamins, glutathione, affect the function of glycans? John, this is a really, really good question. I will see if we have time to answer this at the end, but it might be something that I have to drop you a message about directly. I think we have a lot of partners and a lot of individuals who ask questions about specific interventions and how actually specific interventions affect glycans. And actually, this is giving me the signal that maybe we need to think about doing a webinar on this. So maybe that's one for the next webinar. But I will get back to you, John, on that. So this is, I'm going to show this very briefly. We do tend to show this in a lot of our webinars, so many of you might have seen this already. But ultimately, as I mentioned, glycomics is a huge area of research. And one of the things that we're looking at now is, as I mentioned, people's glycome, so the collection of glycans they have, does change in response to diseases. And so what we're trying to do now is understand actually what is the pattern of change that is associated with various diseases. As you can see, we are filling out this beautiful wheel, which shows us actually what kind of patterns you expect to see with various disease types. But this can be really useful in helping people understand, okay, so, I may have a risk of developing this disease because my glycans are moving in this direction. And so if you're working with patients, you can give them information to say, actually, you seem to have a higher risk of this disease. Maybe we need to think about putting in interventions that help you move you away from that risk factor that you have. And so I think that is a really powerful thing that you can do with clients. And eventually the aim, would be to move into more of a diagnostic tool. And so, glycomics is this really huge area where there's so much information that you can use, really valuable for patients and for clients as well. So as I mentioned, the commercially available glycomic clocks, at the moment, the only commercially available glycomic clock is GlycanAge. It costs $270 at the wholesaler price for partners. So for people who are providing and then doing the consultations, it's done with a at-home dry blood spot test using a collection card. And it ultimately provides information about those pro versus anti-inflammatory glycans. And that information is broken down into various indexes, which can help you understand what risk factors an individual has. Ultimately, it uses the data that we collect to give information about biological age based on the level of chronic inflammation in the body. It looks at potential disease risks as well, and also the most important areas to focus on to avoid some of those disease risks that an individual might have. GlycanAge is the only company that is able to conduct high throughput glycan analysis. So that ultimately means that we can analyze lots of glycans at the same time, which is actually really hard to do. So in the past, one of the reasons why glycomics probably isn't as popular is because it is really hard to analyze them in high volumes. But actually in the lab that we have, we're able to do that. And that's one of the reasons that we probably are the only glycomic clock. And we look at 24 glycan structures that are analyzed, 9 of which are available on the blood spots that we provide. And it's based on a database of over 200,000 samples. And I think that's, to me anyway, really impressive. I think I've spoken about the data sets that each of the different clocks are based on. And I would say that 200,000 samples is the biggest one. And so again, you can see just how deep in the research and how based on the science GlycanAge is. We've just got 2 questions in the chat. I'd like to know the answer to this question too. Okay, so we're getting lots of people who want to know how do different interventions affect glycans. So definitely something that we'd have to do a webinar on. Awesome. Okay, so in terms of benefits, so I think you can see how weighted this webinar was towards epigenetics. And the reason for that is because there is so much information and so many different epigenetic clocks. Glycomics represents a new and a unique way to understand biological aging beyond epigenetics. It can support clinical decision-making for health optimization and disease prevention, which I really think when we think about longevity, that is the aim. It's not about treating disease, it is about disease prevention. But also it can potentially help support disease risk stratification. So what I mean by that is understanding what someone's risk of a certain disease is, and therefore offering personalized advice. But finally, in the future, it could be used for diagnosis because there is so much information and wealth when you look at the human glycome. In terms of limitations, no test is without limitations. And I think with glycomics, what we know is that it's still an area of research. It is not fully understood, and there is a limited number of experts in the field. And I say this because, as I mentioned, we are the only existing commercially available glycomic clock. There are other glyco scientists, but actually our CSO, Gordon Lauk, who is our chief scientific officer, is someone who's been in the field of glycomics for decades, and actually has been doing research in this space for a very long time. But again, because glycans can be difficult to analyze, there are a limited number of experts in that field, and our CSO is one of them. But also, I think understanding why a person presents as pro/anti-inflammatory, despite having a healthy or unhealthy lifestyle, can be really difficult to understand, especially because, as I mentioned, the glycan or the glycome is affected by so many different elements that actually, if you're somebody who has a genetic disposition for longevity, so for example, people who are centenarians who have people in their family who have lived past 100, they may have a genetic predisposition to longevity. And actually, because GlycanAge is a sensitive test to chronic inflammation, that can present in the results. And so sometimes you can get results that are difficult to understand or to explain. And one of the things we always offer with GlycanAge is the education piece. And so, if you are partners who have used GlycanAge before, or you've ever got a result that you don't understand, I really do encourage you to get in touch with us, because we're still at a stage where we like to have personal conversations with people and talk to people about actually what does this result mean, and help people understand how they can actually use GlycanAge in practice. But I do think that is one of the challenges that we definitely do see with GlycanAge. And one of the ways that we're trying to improve that is by giving more information to individuals. Okay, I definitely am way over time, so I'm going to speed through this section and then quickly get some time for questions. So proteomic clocks, I'm not going to speak too much on this because proteomic clocks are less commercially popular than other clocks. They ultimately focus on the number of circulating proteins in the body to help estimate biological age. And I would say it's a more downstream approach. And the reason I say this is because you're not looking at the upstream. You're not looking at the DNA. You're not necessarily looking at the RNA or the genetic expression. What you're looking at is the already expressed protein. And in particular, at the moment, one of the key areas of focus when it comes to proteomics is looking at things like SPAFs, which stands for senescence-associated secretory phenotypes. And ultimately, these are proteins that your body releases when you have a lot of senescent cells, so those zombie cells that I mentioned. And so proteomics is a really interesting field, but actually, I am not aware of any commercially available aggregate proteomic clocks, but there is a lot of research in this space, and a lot of research groups do use proteomic clocks, but they're just not commercially available or for sale. So I'm not going to read too much through this. As mentioned, all of these slides and the recording of this webinar will be provided to everyone. But ultimately, with proteomic clocks, I would say it's a really interesting field. It probably is going to grow. But at the moment, there aren't any companies that have pulled the proteomic data together to create a clock. Many of us will already be using protein biomarkers. So these are things like your high sensitivity CRP, your apoproteins. And even though these are really valuable, they don't quite give enough information to create a biological age estimation. And so my thought or my view is that actually what will happen is people will start to bring this information together to support other clocks, and use that information to actually give more information to the already existing clocks. So just to conclude, we are near the end. I do apologize. I do know I've run over a little bit. When we think about the clocks and how they align with the AFAR criteria, so at the beginning, we spoke about the American Aging Research Group and their criteria of what is an ideal biomarker: predicting future health, monitoring a basic process that underlines aging, repeatable without harm, reproducible, and also works in humans and lab animals as well. So what we know is that epigenetics and glycomics are probably the two that fulfill this criteria best. Proteomics, still a question mark on a lot of those things, mainly because there aren't any commercially available ones. But as research happens in these areas, I do think we will see an improvement in that. And then when it comes to epigenetics and reproducibility, as mentioned, this is a challenge because there are so many different epigenetic clocks. which are based on different areas of the genome. And so actually it's very hard to definitely say it's reproducible because the algorithms can change. And this is why I've put a question mark on the reproducibility of epigenetics. Just to quickly mention this study, which I think is a really, really useful study. So this was an independent study done by the Edinburgh University, and they looked at a number of different clocks. They looked at glycomics, they looked at epigenetics, they looked at even DEXA scanning and various different clinical clocks. And basically, we're looking at how good are these clocks at predicting future hospitalization. So this is an outcome that I think is really important because, of course, nobody wants to end up in hospital. And actually what they found was that GlycanAge was, or the glycan clock was the best at predicting the future hospitalization. So I think that really speaks to how valuable and how able GlycanAge or glycomics is to able to predict disease, but also the outcomes that matter. So just a quick comparison between glycans and epigenetics, because I think these really are the two clocks, to me anyway, that stand out as the forerunners in this space. When it comes to epigenetics, ultimately, because some of the clocks have biomarkers or they look at sites that are not unknown, it is really difficult to understand and analyze them. And I think it's really important that if you are doing tests with patients and individuals, that this due diligence and understanding that element is important, because actually if you're doing clocks that are focused more on chronological age, it's not that useful. You can just look at the person's passport. So really what you want is to find an epigenetic clock which is looking at biomarkers that you know, that you can read about, and that are going to give you the information that you actually want, which is biological age. Again, the question of low accuracy is highly dependent on which companies you're working with and which epigenetic tests you're doing. And so that's something to also be aware of. And then it's not always responsive to interventions. I mean, there are clocks that have proven responsiveness, but again, because there are so many clocks, it's very dependent on which clock you're looking at. When we think about glycomics, again, as GlycanAge, I'm going to speak about GlycanAge because it is the only glycomic clock we're aware of. It is a well-understood biomarker. It's a well-understood biomarker that drives it. There is a high level of accuracy, almost to the point that it is, some people get shocked at just how accurate it can be, especially in responsiveness to chronic inflammation. And also it is, I think one of the biggest pros of GlycanAge and glycomics in general is the fact that it's really highly responsive to interventions because, as I mentioned, glycans are affected by so many different elements of your genetics, your epigenetics, and your lifestyle as well. So, conclusion time. Apologies, guys. I know I'm definitely running over. Just an overview. Aging is complex. It's multifaceted. There's lots of different layers to it. There is no universally accepted single measure or biomarker for aging. It doesn't currently exist. And I think that's something that we all have to accept. Because aging is so complex, it's unlikely that there will be this single universal biomarker. And actually what we need to do is think about aging as a multifaceted thing where there are different ways to look at aging. You can look at it through different lenses. And epigenetics remains the most popular. I would still say it is the most popular biomarker, but is it the best? I think there's a question mark on that. We know there are so many other clocks. I've only gone through three of the categories, but there are so many more. There are lipid clocks, there are multiomics, and there's also the immunome clock as well. If people are interested in learning more about those, please do let us know. You know, we're always looking for webinar topics. But when it comes to my thoughts, both as someone who works with GlycanAge, but also as a clinician as well, I think it's impossible to have one biomarker for the entire organism because of the fact that it is so complex. I think really what you want different ones that give you different views of how someone is aging. But ultimately, because we know that chronic inflammation is the source of all chronic disease, ideally you want to use a clock that is highly sensitive to chronic inflammation and use it as an early warning sign to understand if someone is accelerating and aging faster than they should be. And for me, of course, I would be using GlycanAge to do that because it is an early warning sign for chronic inflammation. But then chronic inflammation can come from anywhere in the body. You know, it could be this person has very fluid osteoarthritis, or it could be they have brain inflammation, which, you know, potentially increases risk of Alzheimer's. And so how do we then use this information that says, okay, this person has more inflammation, to understand how we make a clinical decision that helps them? Well, I think for this, you need to bring in other information. Typically, that would be using clinical clinical basis. So other clinical tests that you can do, and also using history. So like history is probably, I don't know about you guys, but when I was at medical school, it was drummed into me the importance of a history, and I'm really seeing how important it is because it can give you so much information about what might be causing a person's raised inflammation. And then managing the individual with both a generalized and specific approach. Now, I know me saying generalized is going to, people are going to gasp and say, oh my God, how can you say using a generalized approach? I know there is a big focus on personalized medicine, and I 100% agree with it. But the fact remains, there are things that we know work. When we think about increasing longevity, for me, it's the 4 pillars, right? It's diet, it's sleep, it's physical exercise, and it's also your psychological connections as well. And if I think about my experience as a clinician, if I have a patient who's in front of me who presents with a fever and they're really feeling unwell, in general medicine, what you do, if you can't find what's causing the disease, you treat them with what we call a broad spectrum antibiotic to help them get better. And I think when it comes to inflammation, that it doesn't have a specific cause, and actually we're not 100% sure why is this person presenting with this really high level of chronic inflammation with high GlycanAge, it is always going back to basics and saying, well, we know that actually if you improve your diet, you are going to see improvement. If you improve your exercise, you are going to see improvement, and doing some of those things to see the benefit that you want to see, but then also trying to increase the specificness of the approach by doing some further clinical basis testing as well. Oh gosh, okay, so sorry, I had to speed through that last bit. I can see there are 2 questions, but just very quickly, because I think this is important. Actually, this year marks the 10-year anniversary of biological age clocks, specifically looking at epigenetic clocks, so the Horvath clock and also the glycomic clock. And so we're actually offering a 23% discount with the discount code CLOCKS23. So if you are interested in doing GlycanAge tests to celebrate this monumentous 10-year decade celebration of something that I think is really important in the space, then please do take a picture or a screenshot of that code, and you can use it to get a discounted test. And if you are a partner who wants to order for your patients, hopefully you'll have my email address. If not, we'll send a follow-up email, and in that email you can get in touch with us or in touch with me directly, and we can speak about any discount for your patients as well. Oh, okay. I've gone through all of that. I can see there's lots of stuff in the chat. Hopefully people have been answering. I don't know if anyone has any burning questions they want to ask now. I imagine some people are probably don't have time, need to jump off. But if anyone does have any questions, please feel free to drop them in the chat now, and I will take some time now to just answer them. Ah, thank you, Dr. Bruce. Okay. Yeah, anyone feel free to jump off now, but if anyone does have any questions, I will stay on for a couple of minutes to just answer them. Wow, no questions. Did I do that? Oh no, there's a question. We have a chat, something in the chat. Okay, no. Either I did a really, really good job or I did a really terrible job. I don't know which one it is, but I really hope that this was informative for people and gave good information. Okay, I will turn my camera off, but I will still be. Oh, I see there is a question. No. Okay, I will turn my camera off, but I will still be here. So if anyone does all of a sudden think of a question, please do just pop it in the chat and I'll jump back on to answer. Okay, guys, I think we don't have any questions coming through apart from asking about the recording. So, yes, the recording will definitely be provided. If you signed up for the webinar, you'll get an email with the link to the webinar and recording of it. But if there are no other questions, I am going to end the call. Someone has asked, do you provide raw data? Yes, we do. You see GlycanAge is responding. Okay, maybe I'll stay on for a little bit longer. I think some people are all of a sudden asking questions. Okay, guys, thank you so much to everyone who joined the webinar today. As I said, I really hope this was useful for you. If you have any opinions about webinars that you would like us to do, please feel free to email us on partners@glycanage.com with just topics. You know, we're always looking to do stuff that's useful for our partners and people who are interested. So I think it's very clear that the next one needs to be on how GlycanAge responds to interventions. And so we will work on that. Thank you again for your time, guys, and have a great rest of your day. Bye.